I'm actually going to take my headphones off because it's making me feel confused. There we go. Okay, so there's a lot more content on my slides than what we'll be going over just because I was way over time. Okay, so my topic today is on the future of AI in stem cell research. So this is a brief overview of what we'll be going over. And to start, we're going to talk about stem cells. So as some of you may or may not know, this is a creature called a hydra. It's a mythical creature in which if you cut its head off, it will grow 2 new heads from where you cut its head off. And so the question is, how does it do that? You know, what element is there that allows it to regenerate not just 1, but 2 heads? It must be able to still have information to make all the components necessary for a head. And I would say that that is probably the work of stem cells. So what are stem cells? Stem cells are a special class of unspecialized cells that can self-replicate. So when I say unspecialized, what I'm talking about is in the adult body, most of your cells are specialized. They have a specific function that they're highly adapted for performing. And a stem cell is unspecialized in that it's very, very generic. It is the most generic type of prototype of a cell that you could think of. And so being the prototype also means that it has a lot of potential to develop into other cell types. And this is called potency. It's a potency, potentiation. All those variations of the word potent are in relation to its ability to become other types of cells. So the most potent cell is called a totipotent cell or an omnipotent cell. And this cell would be the zygote and early cleavage cells, because the single cell that is the zygote is going to eventually make up the entire human body. So of course it is the most potent that you could possibly think of. The omnipotent cells can make up all cells in the body, but also all the cells and all the components that are needed in the womb. So structures like the placenta and the amniotic sac, etc. Those can be made from totipotent cells. But once you start differentiating, so differentiation is when you start specializing towards a specific cell type. The next class of potency is called pluripotent cells. And pluripotent cells can become any of the cells in the body, but they can't become any of the cells outside of the body that would be in the womb. Pluripotent cells usually will pick one of the 3 germ layers. So in the human body, we have 3 germ layers, as they're called, for anybody who doesn't know. Once they pick a germ layer, after that, they're restricted to fates of cell types that are only available in that germ layer. And then you get more and more differentiated cell classifications as you follow along those lineages. So totipotent, pluripotent, multipotent, oligopotent, and then finally unipotent. So what is the component that allows the differences in specialization? Well, that comes from your genome. So as you may know, all the cells in your body have a uniform set of genes. That's called your genome. And it's the same in all your cells. But the thing is, different cells use and are able to access different genes. And this is called epigenetic modification. So even though all the cells have the same genes available to them, they aren't actually able to access all the same genes. Stem cells are able to access a lot more of the genes than specialized cells are, because as you specialize, what's happening is that these genes get turned off or silenced. And so genes that are irrelevant to the fate that you're working towards get turned off and they're not accessible to you anymore. And so this is an image from the cover of Science magazine in 2010. In the middle, can you see my mouse? In the middle, this would be the zygote. So it's, there's nothing inside it, it's very clear. And so this could be the most potent cell in the image. And then as you move towards the peripheries, you get more and more coloration in the marbles, and you get more and more like stuffed components inside. And that's showing us the differentiation lineages that are available. So stem cell therapy is kind of the big target of doing stem cell research. And this is the application of stem cells for regenerative, repairing, and preventative treatments in medicine. Currently, we only have stem cell therapies available for use in blood-related ailments, but we do have other things in clinical trials. The difficulties in describing a stem cell therapy come from just our general lack of understanding of the differentiation processes. So the first is, if you want to make a certain type of cell, you need to know what progenitor is. Progenitor is basically another word for stem cells. Which progenitor cell line is appropriate to get to that differentiated cell. Then you also need to know what factors, what signaling factors, what signaling molecules are necessary to get that potent cell to differentiate towards the specific lineage that you want and not to one of the other lineages that are available to it. And then finally, you need a way to administer your stem cells to the target tissue such that they get incorporated into where you want them and they don't like move to other parts of the body or just differentiate into what you don't want. So there's a lot of things that have to be overcome when you're trying to prepare a stem therapy. So now we're going to go over the main component of my paper or presentation, which is 3 different papers that use 3 different technologies to streamline the application of stem cell research and stem cell analytics. So before we get into that though, we're going to go over what's called the central dogma of biology. So all your genes are encoded by 4 codes, a 4-letter code called DNA. So DNA is where you start. This is what your genes are. Your DNA gets read, and then it gets what we call transcribed into mRNA. So DNA is a template for making mRNA. mRNA is a messenger that links DNA to proteins, and proteins are the functional end product that is being encoded by your genes. So what mRNA tells us is what genes are actually being expressed. There's a technique called single-cell RNA sequencing, which we can use to sequence all mRNA present in a cell at a single time. So this would tell us all what genes are being expressed in the cell at this exact moment. And then epigenetic analysis tells us what genes are accessible and available for use in the cell at this moment. So there can be a distinction between what genes are actually available and accessible versus what genes are actually being expressed. That can change at different points of time in a cell's fate. We can also use these techniques. We can use them for a single cell and then for thousands of single cells. Or you can use them for thousands of cells at the same time and average that data. So there's a lot of information that we can extract from cells. And the application of AI is to help us get only the most important components from that information. So the first paper was looking at stemness indices in use for cancer. So cancer cells actually can de-differentiate and they can attain more stem-like character. And the problem with this is that the more stem-like that cancer cells are, the more likely they are to metastasize and to move to other parts of the body. And usually, the more stem they are, they're also usually less receptive to available therapies that we have. So in this paper, they used this machine learning algorithm called one-class logistic regression that can not classify, but score cell type identities. So they trained the program on a lot of stem data, so stem cells and a lot of early progenitor cells from each of the 3 germ layers. And they gave it mRNA and epigenetic data. And from this, it was able to identify different markers of stemness in the cells. They then used the output, so the output that they got from the algorithm was 2 for our relevance. We're going to say 2 indices that can score cells on their stem-like character. So the first is using mRNA data, and the second is using epigenetic data. And so they got these indices from doing the stemness training, and then they input data they input cancer data, and they scored all that cancer data. So they had 33 different cancer types with over 12,000 samples of cancer cells. And then they got, for each of these classifications, they got a score of how stem-like this cancer is. So a score of 0 is closer to, is more specialized, and a score of 1 is very highly stem, so more potent. So this is the figure of their output of the 33 classes based on the site is mRNA and the site is DNA modification. And so here on the 1 is more stem-like, 0 is more specialized. And you can read these as box plots. So they give you a range for the approximate value, stem-like values. So here is less, this one is a little bit more. We can also see that the mRNA index and the epigenetic index, they give you different outputs. So there's different information to be gathered from the different sources of information. I just have seen one twice. So what are the applications for these types of indices? Well, the first would be in precision therapies. So in precision therapy, what you could do is you could profile a cancer in your patient against these indices, and get better prognosis for how likely is it to metastasize, what are its treatment, what treatments would be more efficient or less efficient, etc. And we can also use these indices for differentiation therapy. So differentiation therapy is the use of pharmaceuticals, usually against cancers, to induce differentiation towards a cell type that's easier to target and is more receptive to the treatment that we want to use. So in this scenario, we could use the indices trained on the target cell type, and then we could score the cancer after administering the pharmaceuticals to say whether or not or how likely is it to progress towards that lineage that we want that is easier to target. Alternately, we could also use the model itself, not for cancer, but for other types of diseases. We could train it on other disease data and then get scores for tissue samples for how far has this person progressed in this disease, like how likely are they to for it worsening or for getting this disease, etc., those kinds of things. And so the important takeaway of this paper, in my opinion, is this shift from rather than a yes or no disease detection, yes, they have this disease, or no, they don't, is a shift to the scoring of this range. It gives us more information than that might be overlooked when we just ask for a yes or no output. The next paper that we're looking at is about visualization and how we present data. So this is a color spectrum, also called a continuum. And in it, you see a lot of different colors. But the thing is, there's a lot of ambiguity in this figure. There's a lot of ambiguity and indistinctness between where color shifts are and where the boundaries between different colors are. So when is my blue turning into a green? Is this still purple, or is it kind of like a pink now? How do I know? That's very ambiguous. And that's the entire premise of continuous data. It's continuous in that it flows together and it's hard to distinguish how to separate these components. And that's also what we're dealing with when we're working with the differentiation of stem cells. So in this paper, they wanted to visualize the differentiation continuum for blood cells specifically. And so to do this, so here's a little diagram from their paper. They first infected their hematopoietic stem cells, which are stem cells for blood. They infected them with barcodes, and each of these barcodes is unique, so that every single of the progenitors has a different barcode. And then they allowed them to proliferate and differentiate and sequence them at different sequenced samples from the collections at different times. And then they could find those barcodes, and all daughter cells with the same barcode could then be related back to a single progenitor cell. So in this way, they can trace this single progenitor cell, which only had one barcode, is connected to all these different fades, and this is the different routes that they took to get to these different endpoints. So to plot this data, they came up with an algorithm called SPRING, which is specialized for visualizing continuous data. They use k-nearest neighbor classification system, which gives a new data input a label based on its relatedness to already present data points that are nearby. So the input data for the program is single cell RNA sequencing data. The relations between neighboring data points is based on overlap in gene expression. And then the label that is attributed is the expected blood cell fate. So on the left here, this is actually the continuum that they came up with. This is for time overlay. So the peripheries are the later dates and the central darker colors are the earlier samples. And on the right is the cluster boundaries for their KNN outlook model by the end of the experiment overlaid on the continuum. Okay, so this is the continuum again, but with the actual fates overlaid. So here, it gives us a really intuitive visualization of how these fates differentiate and how they relate to each other. So we can see that neutrophils are more similar to monocytes, but they're more different from lymphoids. We also see there's a lot of gray area that is not attributable, and that is not attributed to a single cell fate. And that is, again, going back to this idea of a continuum that is very ambiguous, and that is very indistinct. It's hard to tell when my multipotent becomes oligopotent. Is it this fate or that fate? Maybe it's still bipotent for a really long time. It's very continuous data, and it's hard to make conclusive statements. So the benefits of the spring plot are that compared to older models, it's a lot more intuitive when looking at just the graph by itself. So we can see a spring on the left versus two of the older models, TSNE and diffusion maps on the right. And those two have very not pretty presentation methods. There's a lot of overlap in the lineages. They're not very distinct. It's hard to tell. It's hard to extract information just by looking at those graphs themselves, versus spring is a lot more intuitive. It gives more comprehensible information. Another advantage of spring is that it allows node-specific examination. So this means that if you go back to any of these data points on this plot, you can hone in on that single cell and get all the raw data from its single cell RNA sequencing. And this is great for any kinds of follow-up studies that want to springboard off the results of a prior study, and also great for references for training other types of models that researchers might be using. And on the note of training models, the last paper that we're going to be looking at is all about training models and then sharing that forward. Sorry, I'm losing my voice. So as we know, there's a lot of information that everybody can access on the internet. There's these things called atlases that try to condense the important findings and also the raw data so that's accessible for researchers to use in the future. But the thing is, there's still a lot of work that has to be done before you can actually apply these references to your own research. So in using analytic models, you have to write a code, you have to find the appropriate references, you have to train a model, you have to maybe give it feedback, maybe do some fine tuning. There's a lot. We saw all of this happen with Spring and with OCLR, that you can't just directly apply all this information. So what SC-ARCHES, or single cell architecture surgery, is proposing is a platform for shared transfer learning models that can learn from new data and then be used by other researchers in the future. So they have a lot of models, in supervised, semi-supervised, among some other types. They're mostly artificial neural network type models, and they've already been trained on various cell identification objectives using atlas references that are available to the public. So they have these trained models, they have them available in a repository. Independent researchers can then download a specific model and even select what reference data they want it to be trained on. And then they can use it to analyze their own data. And when they analyze their own data, the algorithm can learn from that new data input. And they can also sometimes adjust and fine tune it themselves. And then they have the option of uploading the modified version of the model back to the repository. And so this uploading of modifying information is called an adapter. And the cool thing about adapters is that you can upload your raw data as another reference source, or you could upload only the weight optimizations that were done to the model as a result of having looked at your data without actually exposing what your data was. And this is useful for, let's say, unpublished research. Let's say you have a lot of research going on, but you haven't published it yet, you can't share that data. Well, you can still share the modifications that were done to the models for other people to use. And the cool thing, okay. So when we look at the model adaptation, it's not just pasting in new data, it's also recalibrating the relations between already present information. So it's more comprehensive. This is a whole other aspect of learning, which is what we call transfer learning. You're adding more information to the system itself. It's not just putting more data points in, it's actually learning from that. And so this allows over time the buildup of fine tuning of models for super comprehensive and super specialized models in the future. But not yet, because this is very new. This just came out a couple months ago. So that'll take some time. What are the applications of SC-Archives? Well, the concept is for this highly adaptable and context-specific models that can be used easily and accessible for all researchers. The proposition or the entire premise of having these pipelined models, so pipeline means forward sharing, which is the forward sharing of information and of the transfer learning to the models. This premise is useful for any discipline or any field that is working with a lot of data analysis, like what we have going on with cell research. So we could use this in spring plots. We already kind of have a similar concept in the OCLR indices, but you can also extend this to further to chemistry, to biochemistry, to maybe some other disciplines that I'm not as familiar with. So going off of all this, what are the opportunities in the future? What jobs or emerging occupations does this bring with the development of the field? Well, I think there's three important bases where opportunities are emerging. The first would be for programmers. So as we saw, there is a need for highly adaptable analytic algorithms, especially for highly focused research needs. So this would be for research that is either highly specialized, maybe it's an emerging field and there's very limited reference data, and maybe available programs just aren't precise enough, aren't accurate enough for the future, for really getting conclusive results. So this would be where you would have a need for programmers. To make those types of after making those types of algorithms, we also need people who can interpret that. So the next step is for bioinformatics interpreters in the lab. So this would be people who can understand the data output of algorithms, understand that what it's implying for the research results, and then also provide the programs with feedback and with fine tuning where it's needed, especially for supervised models that are all about getting feedback about are they right or are they wrong. Let me adjust my understanding of all the components. And then, and then the last platform is again for bioinformatics interpreters, but this time in a clinical setting. So for ongoing clinical trials, I think there would be a need for informatic interpreters who can understand emerging findings from emerging research and then translate the implications of those findings for ongoing research. How can we adapt this new information for what we are already working with? And then eventually in the future, when we have more therapies in practice, that we would also need these professionals in the precision medicine field to, again, understand emerging findings and emerging treatment styles, and then transfer this to ongoing treatment plans and ongoing treatment counseling for patients who are already ill, and how we can best utilize these emerging technologies and emerging treatments for who we are working with. So in conclusion, we talked about STEM. We talked about 3 different platforms for streamlining data analysis. And we talked about some future prospects. Do you have any questions? Also, here are my references.
Okay. Are there any questions?
I'll just share this in case we need the slides again, actually. Yes.
Yeah.
This is actually very related to a research to a research capstone I'm currently doing. We're actually trying to make a pipeline as well to use single cell data. I was just curious. You said it could be applied to like other fields. Is that based off of the way that it was designed?
So not specifically this, because this was specifically designed, again, they trained it on singles on like cell data and cell identifying objectives, but the concept of having this platform with models that can be shared, that would be useful for other disciplines.
Okay, so this isn't like the actual pipeline that's generalizable to other fields.
Not to other fields, yeah, this would just be for cells.
Okay.
Okay, any other questions? So we could go on then to Matthew. Yeah.
Okay.
So it's the same deal. I think we should turn off these and you should do.
Is this showing up now? I think if you switch to speaker view, it should. Oh, really?
It has a definite video. Well, we can show them all and then you could.
I just closed mine.
There.
Okay. Okay.
We're good.
Perfect. Okay. Well, hello everyone. Can everyone hear me online?
Yeah.
I'm sharing my audio. All good. Oh, okay, they can hear us. Okay, I'll get started.
Yeah, no, we can hear you, but why aren't we seeing your slide?
Oh, this is just the first one. Just not to ruin the first one, yeah.
It's a totally white slide, eh?
Yep.
Oh, that's all right.
Okay, imagine you're walking through the hallways of Eka when suddenly a genie appears from a seminar room and grants you 3 wishes. What would your 3 wishes be? This time of year, you're probably only wishing to ace all your finals. Come the new year, perhaps you would have more health and fitness orientated wishes. When you think of how drastically your life could change, given the opportunity to achieve peak mental and physical states, you're not likely to turn the genie down and carry on with your life, right? Well, would you turn down that same genie if it came in the form of a computer and prosthetic limbs? It's something to think about, at the very least. My name is Matthew Pletz, and today I will be discussing brain-computer interfaces and prosthetics from the perspective of biomedical engineering. The countdown to the technological singularity is on. One of the ways that the singularity may be achieved will be through transcending biology with the use of advanced brain-computer interfaces and prosthetics. The recording and functional usage of electrical activity generated by the brain is an area of investigation in which many fields, including biomedical, is the screen okay? It's kind of half of my presentation.
There we go.
Oh, okay. Sorry, I just realized that now. Okay. Sorry about that. Well, it's fixed now. Where was I? The recording and functional usage of electrical activity generated by the brain is an area of investigation of biomedical engineering as well as many other fields. It is a field that resembles science fiction as much as it does medicine and engineering. In 1929, German neuroscientist Hans Berger discovered the electrical activity of the brain using EEG. In 1929, it is predicted that the thinking abilities of recently deceased individuals will be transferable to computers by the year 2090. This could result in an immortal human brain. The time from the first EEG recording to fully uploadable conscious is estimated to occur within a 160-year period, and this is a conservative estimate. It could be quite a lot shorter. In this presentation, I aim to investigate the present-day uses of brain-computer interfaces, the future of BCIs and prosthetics, as well as their resulting impact on humanity and medical ethics. So, how can we make predictions about the future? Perhaps the best way is to look back to the past and see how it has influenced who we are today. This is one quote that I've found really resonates with me by David McCullough. He said, "History is a guide to navigation in perilous times. History is who we are and why we are the way we are." So, where are we today? That brings us to Chapter 1, present day. Brain-computer interfaces have recently received widespread attention in the media due to the announcement of Elon Musk's startup company, Neuralink, which plans to provide brain-machine interfaces to connect humans with computers. BrainGate of Cybernetics is a current BCI that acts as a communication device which converts the activity of the central nervous system into a signal that can be utilized in order to perform a variety of tasks, including augmenting or repairing cognitive or sensory motor functions. This can be achieved through a number of different mechanisms. Stimulating muscles to regain lost function, such as the ability to speak or move, is one method of regaining control of the body. Many techniques exist to measure brain activity that differ in regards to invasiveness. Most BCIs, including the BrainGate system, have utilized an invasive approach in which electrodes placed on the surface or within the cortex detect electrical signals of the central nervous system. Non-invasive approaches include EEG, applying electrodes to the scalp, and BCIs that use metabolic activity that is measured through functional magnetic resonance imaging. Invasive implementation is performed to record the electrical activity of the CNS with greater accuracy as compared to non-invasive modalities. Motor hand function originates in the superior part of the precentral gyrus. Microarrays are implanted into this precentral gyrus precisely in the primary motor cortex arm area, located on a knob-like structure. This structure resembles an epsilon on the axial plane and appears hook-like on the sagittal plane. Therefore, it is visible on the cortical surface as it sits between the precentral and the central sulci. Motor impairment resulting from an inability of intact movement related centers of the brain to generate movements due to damage to the spinal cord, muscles, or nerves is an affliction that affects hundreds of thousands of people. Neuromotor prosthesis, NMP, in the motor areas of the brain. Currently, the clinical populations that are best served by BCIs control prosthesis include those severely disabled by disorders such as ALS, cerebral palsy, brainstem stroke, muscular dystrophies, or chronic peripheral neuropathies. One of the most moving case studies I've researched was a case involving Jan Arden, a tetraplegic who, for the first time in 10 years, was able to feed herself due to a BCI implant. In this study, Jan, who was diagnosed with spinocerebral degeneration, was implanted with an intracortical microelectrodes and trained to operate an anthropomorphopoietic prosthetic limb. Within 2 days of training, the participant was able to freely move the prosthetic in a three-dimensional space. After 13 weeks, the participant was able to use the prosthetic limb to do skillful and coordinated movements that resulted in clinically significant gains in tests of upper limb function. So, who can benefit? Generally, people with ALS, severe cerebral palsy, and brainstem strokes retain very limited, easily fatigued, and/or unreliable eye movements, or minimal muscle function, and thus cannot be adequately served by conventional muscle-based communication technology. With the present-day functionality of BCIs, these people may be able to express basic communication and control that is more reliable than existing technologies. For these people, brain-computer interfaces may result in significant improvements to their quality of life. A shortcoming of current devices is biocompatibility that results in reductions to prolonged effectiveness. A significant decrease in the number of recorded waveforms occurred starting approximately 6.5 months after implementation of the device into a participant of a 2004 study. Gliosis and other biological events result in a reduction of effectiveness. Another shortcoming of present-day BCI use is user functionality. Studies are confined to a laboratory setting. This results in participants not being able to use the technology in day-to-day living. Additionally, experienced technicians are needed to manage the system, making it impractical for use in a non-professional setting. Now that we have explored the past and the present, let's move forward to Chapter 2, the future. Currently, the majority of brain-computer interfaces are targeted at restoring or improving the quality of life of individuals with neurological disorders and/or paralysis. Future uses of brain-computer interfaces and prosthetics may fundamentally change how the general population interacts with the world. This may include changes in military strategy, professional environments, and socialization. The Pentagon has awarded funds to a Defense Advanced Research Project Agency, shorthand DARPA, backed brainwave research project called Silent Talk. This project aims to map EEG patterns to individual words and to determine if these patterns are generalizable, given there are similarities in the recording of the EEG patterns. The U.S. Forces' goal is to allow soldiers to communicate silently, using only their thoughts. In 2018, DARPA announced that a participant with an experimental brain-computer interface used their mind to simultaneously control 3 aircraft. Additionally, the Cognitive Technology Threat Warning System program plans to use EEG patterns to create mind-reading binoculars that will alert the soldier to threats in their environment faster than the conscious minds can interpret them. This function would be achieved by the interconnection between a brain-computer interface and artificial intelligence, by utilizing the advances in flat-field wide optics and large pixel count digital images. DARPA believes that a breakthrough in this program will be imminent in the near future. Not only may brain-computer interfaces be used to control and be used in communication, but also involve improving physical performance. A BCI system may enable the mitigation of pain via pharmaceutical distribution. It could also involve improving strength through more efficient integration with mechanical exoskeletons. The exoskeletons of the future may not necessarily be the bulky armor-like suits that are often portrayed in media, but rather soft wearable material that resembles a second skin. The advantages of this type of exoskeleton is that it will allow the user to retain full range of motion and will fit comfortably like an item of clothing. These exoskeletons will likely use pneumatic muscles that produce similar forces to electrically powered motors, with the benefit of being significantly lighter. Flexible exoskeletons may allow the soldier of the future to generate the force needed to lift several hundreds of kilograms. In addition to military applications, brain-computer interfaces may also be used in the civilian workforce. This may be used to fundamentally change the workplace by interacting with machines and/or be used in high-risk environments. At a basic level, brain-computer interfaces may be worn by surgeons and pilots to monitor drowsiness levels. One way that BCIs may radically change the workplace is by allowing access to individuals otherwise unable to work. In the United States alone, there are more than 40 million Americans who have a disability, making up over 12% of the US population. Moreover, according to the Bureau of Labor Statistics, only 19.1% of those suffering from disabilities are employed. With the current trend of increasing automation and reliance on artificial intelligence, the employment inequality for individuals with disabilities is beginning to shrink. Telepresence, robots that receive input from humans and outputs to robots anywhere in the world, is another method that is currently being used to allow for greater access to the workforce, as well as healthcare. So one area that's pretty interesting of the application of telepresence in medicine is telesurgery. So telesurgery is the long-distance surgery, which can allow access to people without having to travel and conserve populations in remote communities. This is currently becoming a reality and may one day become the norm in hospitals. The origin of telesurgery has roots in the space race, actually, with the question being, during human spaceflight, how would astronauts be treated if several thousand kilometers away from an operating room? In the 1970s, NASA began exploring remote-controlled robots to operate on astronauts. In 2006, the Zeus robot simulated a spaceflight telesurgery at the Aquarius underwater base. More recently, the Da Vinci system has been introduced as the first market-ready robotic surgeon. One surgeon embracing this technology is Dr. Mehran Advani, who uses the Da Vinci system to work in a community hospital that lacks the facilities of his hospital in Hamilton, Ontario. The ease of use is so great that to Dr. Advani, it is basically the same as if he were next to the patient, just using telecommunication and robotics. It doesn't feel different, he says. Telesurgery has an ability to resolve limitations of laparoscopic procedures as well. Although beneficial to the patient by reducing invasiveness, laparoscopic procedures have resulted in few benefits for surgeons. The surgeon loses a sense of the hepatic feedback, a clear three-dimensional view of the surgical field, and experiences reduced dexterity due to the limitations of the instruments. Increasing use of telesurgery may offset some of these limitations inherent to that laparoscopic procedure. A surgical robot may allow for a magnified stereoscopic image that equals and may even surpass the visualization encountered in open surgery. Additionally, the dexterity of the surgeon may be improved using the robotic system. In the future, advanced algorithms may allow these robots to go into an autopilot mode and finish the surgeries. This would allow for surgeries at a great distance, for example, from Earth to Mars, without the concern of system lag or delay. With the rise of commercial spaceflight, resulting in an increase, I guess it increases the possibility that someone will be in spaceflight and actually needing to undergo a procedure as more and more people are beginning to travel to space. So in order to account for the complexities of surgery, developments of remote-controlled surgical robots that utilize artificial intelligence are needed. Now that we have envisioned the future, let's take a few moments to ponder this future. In Chapter 3, Ethics. Surpassing innate biological limitations of the brain will be a potential mean in which humans transcend biology. In the future, brain-computer interfaces may be used as a mechanism of bionic enhancement of normal cognitive, memory, sensory, or motor function, memory augmentation, infrared vision. These augmentations will generate many ethical dilemmas, including the creation of super soldiers, cognitive and physical changes enhancing athletes' abilities beyond human limits, affordability and equitable usage of scarce resources in healthcare, and inequality of access to implants, especially in developing nations, and commercial influence exerted both on the public, scientists, and physicians. A concern for the soldier of the future is addressing the capacity of the participant to give meaningfully informed consent to undergo procedures to enhance their abilities. This may be difficult in the secretive nature that militaries often work around. Additionally, the hierarchical military structure can result in pressures for soldiers to comply with orders from their superiors. The decision-making process of the soldier may also be impacted by the enhancement procedure. For example, a brain-computer interface may be used to inhibit a soldier's capacity to make moral decisions. This poses the risk of misuse for those with control. The soldier's sense of self and identity may be completely altered. Outside of military use, brain-computer interfaces may have the ability to transform what it means to be human. This vision of the future is summarized by Riley Green, a brain-computer interface clinical researcher, who says that it has become increasingly evident that neurotechnologies have the potential to profoundly shape our human existence and sense of self. The sense of self may be radically changed in the future, where brain-computer interface private companies have access to the neural data of their users. Furthermore, questions arise as to how EEG data may be used in order to infer emotional states and cognitive processes, which would provide unparalleled insight into user functions and intentions, preferences, and emotions. In a dystopian future, similar to that envisioned by George Orwell in the novel 1984, a totalitarian regime may collect brain-computer interface data in an effort to exert social control through mass surveillance. In order to prevent this future, there will need to be extensive revisions to the Charter of Rights and Freedoms in order to protect the people who utilize BCIs. This is currently already occurring in Canada. Canada's system of justice is modernizing the Privacy Act in order to address the dramatic technological changes that have occurred since the original policy came into effect in 1983. A modernized Privacy Act may help to support the protection of personal information held by the government. Individuals expect better digitally enabled services and results while their personal information is protected. In addition to the privacy concerns, regulators of the future will also need to address concerns related to misuse and unequal access. Disparities may also be exacerbated by BCIs when used for cognitive enhancement. When used for academic or professional advancement, there may be unequal access due to existing social inequalities. The question becomes, is this the future that we want, and how will it shape our understanding of what it means to be human? To researcher Riley Green, despite the potential risks, the ability to integrate the sophistication of the human mind with the capabilities of modern technology constitutes an unprecedented scientific achievement, which is beginning to challenge our own preconceptions of what it means to be human. With increasing use of telesurgery, there are ethical and regulatory concerns that need to be addressed. Increasing medical costs, especially in the United States, may result in a new form of medical tourism. Patients who are otherwise unable to afford the healthcare provided in their country of origin may undergo procedures performed by surgeons operating outside of their home country. This poses the question, in the case of an adverse surgical event, who would be responsible and who would be liable, and how would this telesurgery be regulated? For telesurgery to become prevalent in the future, universal standards to address and prevent maleficent telemedicine will be needed. Additionally, even if the technology becomes widespread, would people embrace it? This question may be asked by both patients and physicians, as telesurgery will fundamentally alter the physician-patient relationship that exists in medicine. Telesurgery has raised ethical concerns because it has been suggested that the predominant threat of technology is the tendency to dehumanize persons, manifest through a reduction to objects. The personalist perspective in medical ethics suggests that the most meaningful care for the patient is achieved when the patient is viewed according to all their dimensions as a person. If current practices of care avoid reducing the patient to an object, then it must be ensured that any new medical technology introduced does not threaten to diminish patient care. Additionally, will the technology of telesurgery be available to underdeveloped countries and rural communities? In theory, yes, but given the need for advanced telecommunications and healthcare infrastructure, it is possible that the technology will not exist outside of developed nations. In conclusion, current-day usage of brain-computer interfaces and prosthetics aims to regain lost motor and neurological functions. The ability of such devices has a potential to change future patients' quality of lives, as well as the field of rehabilitative medicine. However, there are many barriers that need to be overcome before widespread use. These barriers include EEG sensitivity of measurements, as well as improvements in biological compatibility. The future brain-computer interfaces and prosthetics may have a remarkable impact not only on the future of healthcare, but also may dramatically change how humans interact with one another and the world. In the future, BCIs may be used for a variety of military, civilian, and social and medical ventures. Given this, considerations of privacy and legal structures will need to be undertaken to protect future users. Given that BCIs may also have the ability to fundamentally change how we interact with the world, it will be necessary to consider the ethics of how they may change us and what it will mean to be human in the future. So society is on the path to the technological singularity, whether we're prepared for it or not. And in order to better comprehend our impending future, we need new ways of promoting discussion about the technological future. And Dr. Soles and this course were one inspiration for the creation of an original poem, which I will share with you now. Future Self. Once I was alone, before this computer and I became one. Since then, oh, how much I have grown. It lets me lift metric tons by these artificial limbs. And don't forget how much I can learn. Knowledge and strength, you don't even need the gym. How could I concern? Sure, Big Brother knows, privacy is gone. Some feel the need to shout, whoa! Perhaps let's evaluate first. Hold on to the state of oneself, where you truly are yourself. And I know what you're thinking, and don't worry, I will be quitting my day job to become a poet anytime soon. And then just to finish off, here are my visual references, as well as my references to my ideas of the presentation. Thank you very much for listening.
Yeah, so you can turn off the sound system on your. Turn on the microphones in the room here. Yeah, so one thing that I realized listening to you is that there's going to be a kind of interim period when it seems like thinking about everything on a spacecraft as being something with medical purpose isn't going to make much sense, right? Like the Da Vinci surgical operating system is going to be big and bulky. And what if nobody gets sick and needs an operation? So they're going to need dual purposes. Those machines that can operate on human beings need to be capable of doing some other things too, because otherwise people will complain about using such a huge amount of space for something that, the whole mission they didn't ever use, right? But then later on, probably they'll become more compact and somehow this dual use won't be necessary because the thing is so compact anyway. But I can see during an interim period that, astronauts, for instance, might complain that, physicians had too much influence over what we have in here. And when nobody's sick, half this stuff isn't being used, right? So that'll be, yeah.
Yeah, that's an interesting point. I think, ideally, if moving forward, we can introduce a system that has a bit more functionality and it's not just limited to being a surgical instrument, but also has other mission-related functions. That will be the best way that we can introduce that system immediately without that initial backlash. But I think given just the history of humans, we always say we don't need something until disaster strikes and we realize that actually, this is what we need. The same thing was said about the Titanic and lifeboats, oh, we don't need lifeboats, they're a waste of space. But, when you see the consequences of not having them, then it becomes just that much more prevalent as to why, something that you might not need all the time is necessary to have all the time.
Yeah, I think it's an important trade-off. And I'm quite proud that thus far, none of these presentations have been way less interesting than others. You could think how bad you'd feel now that we've done 6 presentations if you gave the one that was really boring. No, they've all been right up there, you know, and the same with your papers. That like I'm used to papers with a lot of typos and, you know, things that need to be, you know, corrected in a, and thus far your papers don't have that, which is kind of cool, you know, that they're not like major spelling errors and things. So what does that mean? It means the average quality of the student in the course continues to go up, you know, and you're sort of setting a new level for, you know, performance, which that's also cool. And it's kind of how things should, but both things should happen. I should be constantly improving the course and adding things to it, right? So you don't want to think that the bells and whistles in the course today are just what we had in, you know, January 2012, and we have not added anything. But that's kind of interesting. If you think about the videography techs, so in the beginning, we had 3 full-time techs in the same room with us. It was really cool. But right after that, the university fired most of those people, you know, part of the downsizing and so on. So it was really sad. Yeah, but anyway, so yeah.
I have a question. On the topic of the mind reading for the soldiers. So I thought the premise of the first part was we train the AI to read brain signals to different words so that they can interpret words that are being thought, right? But then you said the application of that for mind reading of other people, of like enemies.
Sorry, that might have been a bit of miscommunication on my part. So the idea is that it can recognize patterns through the actual binocular device, and then using AI to create movement recognition patterns to then predict, I guess, where the combatants or where the individual they're looking at is going to move. So it's both a predictive model, but then also being able to react to even just small changes of movement that, you know, the human eye might not be able to pick up on, and then being able to alert the individual who's using that device.
Okay. Yeah, I got confused that I was mind reading the other person, but it's actually like visual reading.
I believe so, yeah.
So it's sort of the AI Wayne Gretzky, right? What Wayne Gretzky was good at was predicting where the puck was going to be, right? But if you think about it, probably AI would be quite good at that too. You know, figuring out, well, a few seconds from now, it'll be here. Yeah, yeah. So that's part of the predictable future. Okay, so other questions? Yeah, I'm quite pleased at how all 6 of these presentations thus far have gone. And so what I would suggest, just because it's convenient for me, but let you know, let me know if you have a huge pushback to this. So I have a grant that's due January 5th. So I'm kind of thinking creating the individual videos for you guys, I would do after January 5th. So after that point, you'll have your own video, not contaminated by any classmates. It's just you throughout, right? But until January 5th, you won't. So you'll, like, you know, today will contain 3 presentations, the previous one also. That just sort of makes sense to me. That I'm willing, since I'm the guy doing the editing, I kind of like to do that when my big, you know, thing that I'm focusing on is sort of over. Yeah. So you have that to look forward to after you're gone from the course. So like a week or so after that, you'll find that suddenly there's a video of your final presentation that is just you, that is edited down to being just you. So I'll do that in early January. Unless some student says, no, that's so wrong. You need to do it now. I don't want those other people in my video. Yeah. So if one of you complains like that, I'll have to shift my plan. Yeah. Okay, any other questions? I think it's kind of cool. Our visitor from India, yeah, let's see, she's still here. But anyway, she has decided that she wants to, she's going to take the whole course in the next term. So not only go back and sort of revisit the past year of it by binge watching the course, but also start taking it in January again. So yeah, so at least you convinced one 14-year-old in India that this content that we're creating is really watchable. Okay, so I guess that's it for today. Thank you all very much.